Hello, welcome back. My name is Larissa, and I am a senior embryologist studying to take the TS board examination. Welcome to Lesson 2, Part 2 of the Art Compass Academy. Today we'll discuss embryo culture media and systems. Previously, you learned about the importance of pH in biological systems, how pH is maintained by buffers, and common ways to measure pH. Today we will cover media used in an IVF lab, media composition, and the nutrient and pH requirements of cells and tissues. An embryo culture system is the specific combination of environmental conditions, technical equipment, and particular nutrients that embryos require for optimal growth. Perhaps the most important factor is the embryologist's ability to maintain a stable environment within a laboratory. In order to optimize this environment, we must tightly control all conditions affecting the embryo, including light, temperature, humidity, and the concentration of gases that each embryo is incubated with. The media used in culture systems varies widely and can include sperm preparation, handling, and freezing media, oocyte retrieval media supplemented with protein, and occasionally heparin, embryo fertilization, cleavage and blastocyst stage culture media, and vitrification and thawing media. Lastly, light or heavy oil, and wash buffers that are used to rinse plasticware, pipettes, and oil, for vaginal and cervical washes, and to wash biopsy trophectoderm cells. Modern embryo culture media was originally designed to deliver the proper nutrients to the embryos when they need it the most sequentially. For example, a day 1 embryo does not use a lot of glucose as it develops initially. But on day 3, as the embryo starts to make its transition from a multicell embryo toward a blastocyst, the embryo's utilization of glucose goes up significantly. So on day 3, the embryos are moved to a media that has a greater concentration of glucose. Newer, single-step culture media has been designed for uninterrupted culture, such as for use with a time-lapse incubator. The impact of sequential versus single-step media on obstetric and perinatal outcomes are still being investigated. Regardless of how media is used, all media has core components such as salts, buffers, energy substrates, essential and non-essential amino acids, a chelator like EDTA, and antibiotics and pH indicators. All of the previously mentioned components must be dissolved in a solution. Osmolality is a measure of the number of dissolved particles in a fluid, and it is as important as the pH of culture media. It has been known for decades that mammalian embryos develop over an osmolality range of about 255 to 295 milliosmoles per kilogram. When osmolality in culture media exceeds 300 to 310 milliosmoles per kilogram, it might impact the embryo's viability and inhibit embryo development. If evaporation is occurring under the culture conditions that are used, then osmolality will increase each day over time. The specifics of the culture system, such as humidity, oil density, affect evaporation. Most IVF labs still change media on day 3 to avoid excessive evaporation, because the media will experience only 3 days of evaporation instead of 5 or 6 days. The intracellular pH or pHi of oocytes and embryos plays an essential role in maintaining homeostasis and regulating several critical metabolic and transport processes, including protein conformation and glycolysis. In human embryos, the pHi initially follows the external pH of the culture environment or pHe, and any fluctuations in the pHe will affect the embryo's development. In general, a physiological range of 7.2 to 7.4 is aimed for in culture media. The pHi of early human preimplantation embryos appears to be regulated by opposing mechanisms: a bicarbonate chloride exchanger, sodium-hydrogen antiporter, and possibly a third acid alleviating transporter that was both sodium and bicarbonate dependent, resulting in the maintenance of pHi within a narrow range. Embryo culture systems were developed to mimic the environment found in the female reproductive tract. By creating an environment that closely resembles the natural circumstances of conception and development, we hope to give embryos in a laboratory the same advantages available to them inside the human body. Thank you for listening, and don't forget to download the Certificate of Achievement to submit for CUs.